Hello, my name is Sam Jimenez, and today we'll be learning about Ehlers-Danlos syndrome. By the end of this presentation, you should be able to describe differences between the 4 types that we discuss, identify at least one genetic mutation that occurs, and describe the process of collagen synthesis. First, we'll start with an overview about what Ehlers-Danlos syndrome is. So this encompasses a group of genetic connective tissue disorders that are characterized by defects in collagen synthesis or structure. EDS is characterized by a variety of symptoms, including joint hypermobility, skin hyperextensibility, and fragile tissues. And these conditions are caused by mutations in genes that are responsible for collagen production or structure, which is crucial for the strength and elasticity of connective tissues. There's 13 types of Ehlers-Danlos syndrome, and each type is associated with specific genetic mutations that affect different aspects of collagen synthesis. But here we're going to explore 4 major types, which include classical, vascular, kyphoscoliosis, and hypermobile. And we're going to focus on gene expression differences and how they lead to their distinct phenotypes. So first we're going to dive in to the world of connective tissues. So connective tissues are tissues that play a vital role in our bodies by providing support, connecting organs, and maintaining structure. One of the key players in connective tissues is collagen, which is a strong and fibrous protein and the most abundant protein in the human body. Collagen is responsible for giving our skin its elasticity, strengthening tendons and ligaments, holding our joints together, and even maintaining the structure of our blood vessels. There's 5 main types of collagen that each play a different role in the development throughout the body. As you can see here, type 1 is structure to skin, bones, ligaments, and tendons. 2 is joints. 3 is muscles. 4 is skin. And 5 is cornea. And type 5 helps with the cornea, hair, and tissues of the placenta. It's important to note that lysyl hydroxylase also plays a role in collagen formation, and this basically helps sustain the extracellular matrix. Now, before we begin discussing Ehlers-Danlos syndrome and its impacts on collagen, we need to understand normal collagen. So collagen synthesis begins with the transcription of specific genes that encode for these collagen proteins. There's many genes involved, for example, the COL1A1 and COL1A2 genes, and the genes are typically located on chromosomes and contain the genetic information for collagen production. So once the collagen genes are transcribed into mRNA molecules, these molecules will undergo processing in the nucleus, and this can include capping, splicing, and polyadenylation. And this basically forms more mature mRNA, and this contains the instructions for building the collagen proteins. This mRNA will then leave the nucleus and enter the cytoplasm, and ribosomes will bind to it to initiate the process of translation. So during translation, amino acids are brought to the ribosome by transfer RNA molecules, and amino acids are linked together to form a growing polypeptide chain, and that chain is going to be according to the mRNA sequence that was provided. So as this polypeptide chain continues to grow, it will fold into specific 3-dimensional structures, and then it'll be inside the endoplasmic reticulum where the polypeptide chains are modified and undergo hydroxylation of those amino acids by enzymes such as that lysyl hydroxylase that was discussed. And these modifications are crucial for stabilizing the collagen triple helix structure. So these modified polypeptide chains are now called procollagen and will undergo further processing and folding, and eventually will lead to tropocollagen being formed. And this tropocollagen molecules will be what forms into the collagen fibrils in the extracellular matrix. And that will also occur via lysyl oxidase, which is going to mediate the cross-linking between these fibrils. And once this cross-linking happens, it's going to basically contribute to the strength and stability of collagen fibrils, and then it's going to be organized into larger collagen fibers in the tissue. And as you can see here, this is also the step-by-step of the collagen synthesis. So the next thing that we need to discuss is transforming growth factor beta, or TGF-beta. And this is a type of signaling molecule that regulates various cellular functions, and it's going to play a crucial role in controlling the growth, proliferation, differentiation, and apoptosis of many different cell types in the body. But for collagen, TGF-beta has an important role in its production. And basically, it's going to stimulate the initial collagen synthesis by upregulating genes such as that COL1A1 and COL1A2 genes. And it's also going to initiate the fibril formation and regulate the cross-linking that happens between those collagen fibrils to then make the bigger collagen fibers. And finally, TGF-beta has an important role in tissue repair and remodeling, which is very important in Ehlers-Danlos patients because they typically have impaired wound healing abilities, and that's due to this mutation in this pathway. So overall, these tissues lead to Ehlers-Danlos syndrome because there's a mutation in genes within the collagen synthesis pathway that leads to abnormal collagen development, and this is going to be a disruption of collagen fiber formation, which leads to the symptoms that we discussed, such as the hyperextensibility. It can also lead to weakened blood vessels, which predisposes patients to ruptures of those blood vessels. In the image to the right here, on this A side, we can see normal collagen formation, and the B side is collagen from a patient with classical Ehlers-Danlos syndrome, and you can see the differences in structure. So for classical Ehlers-Danlos syndrome, it's typically going to be an issue with the type 5 and type 1 collagen. For hypermobile, it's still unknown. For vascular, it'll be the type 3 and type 1 collagen, and for kyphoscoliosis, it's going to be mutation of that lysyl hydroxylase that we discussed. So next we're going to discuss the mutations. So as previously mentioned, the different types of Ehlers-Danlos syndrome are caused by different mutations in various genes. And this figure kind of shows the division of those for classical, vascular, and hypermobile. But first we're going to discuss classical Ehlers-Danlos syndrome. And so this occurs by mutation in the COL5A1 or COL5A2 genes. And the mutations are typically either missense, which is a single nucleotide gets changed and results in a codon change, or exon skipping, which is when part of the DNA gets skipped over, and this can lead to a shorter protein being formed. So mutations in these genes are going to impact the type 5 and type 1 collagen, which are responsible for the skin, hair, ligaments, and bones. And it's overall going to lead to an issue in fibril formation and stability and impair that collagen formation. And it's important to note that this is what contributes to that elastic skin and the typical symptoms that we see, as well as joint instability. And as far as environmental factors, so Ehlers-Danlos syndrome is genetic. It's autosomal dominant. However, there are environmental factors that can exacerbate these symptoms. And for classical, this includes ultraviolet radiation because it causes issues in the cross-linking pathways. So for hypermobile, there currently is not a known gene that causes hypermobile Ehlers-Danlos syndrome. There's speculation, and due to the symptoms present, researchers can conclude that it is a gene in collagen synthesis, but exactly which gene is still unknown. For kyphoscoliosis, there is typically a missense mutation in the PLOD1 gene, which encodes for that lysyl hydroxylase that we discussed, and so therefore impacts the cross-linking ability. And this will compromise the structural integrity and strength of connective tissues. As far as environmental factors go for this, nutritional deficiencies can also exacerbate these symptoms. And overall, it's just going to impact that lysyl hydroxylase cross-linking problems in the collagen synthesis pathway. For vascular, there's typically a problem in the COL3A1 gene, which codes for the type 3 collagen. And this is going to cause a decrease in the structural integrity of blood vessels and organs, and this is very dangerous and life-threatening because it can lead to dissections and ruptures. Some environmental factors that can affect this is anything that can impact your vascular health, which includes smoking, consumption of alcohol, as well as certain medications. Here in this figure, we can see all of this summed up, so exactly which genes are impacting the different types of Ehlers-Danlos syndrome. And of course, hypermobile is not discussed because, again, we do not know the gene. So I hope that you learned something from this presentation, and thank you for doing it.